Can you hear me? Let me know in the chat down below, please. We're gonna be starting very soon. We'll do the quiz together, and if you have any questions, stop me while doing it, and I'll explain it. Okay, I'm gonna wait just like 2 minutes, just so everyone can join, so we don't have to repeat. Okay, I don't mind repeating though. Okay, 1 more minute. Thank you, Zainab, for joining us today. Okay, Yola, let's begin. So the first question, which of the following are true about stem cells? Remember, we said stem cells are unspecialized, undifferentiated cells. They produce daughter cells that can become specialized, we said, through mitosis. And we said there are two types, embryonic and adult stem cells. Now, why is the fourth one wrong and the fifth one wrong? Remember, we said embryonic stem cells have a wider range of differentiation. And of course, the embryonic stem cells do not continue to exist because as we grow older, more cells are specialized, more cells are differentiated. We do not have more embryonic stem cells. Let's go next. The table compares between mitosis and meiosis. Which of the following is false? So it requires only one nuclear division. It requires two nuclear division. That's correct. Produces two daughter cells. Produces four daughter cells. That's also correct. Produces daughter cells with the same number, exactly, because remember we said diploid to diploid, while in meiosis we said we go from diploid to haploid, so they have half the number. Produces daughter cells that are genetically identical, that is correct. In meiosis, however, they're not genetically identical. Remember we said two processes that cause genetic variation, which was crossing over and independent assortment. So results in diploid body cells results in haploid body cells. So this question, I think it's a bit iffy because 5 should be correct. Meiosis does result in haploid bodies. Oh, maybe because body cells is incorrect. It should be gametes. Okay, so maybe that's what they were trying to test you on. Let's go next. Which of the following is not a function of mitosis in animal cells? Remember, we talked about the functions of mitosis: produce new cells and grow. That was one of them. It also converts a single celled zygote to become a multicellular organism. It also participates in replacement of worn out cells and also participates in the repair of injured body parts. Exactly. Meiosis produces gametes. For meiosis, it's gametes. Yes, meiosis produces gametes. Gametes are basically sex cells. Okay, that's the purpose of meiosis. Let's go next. Okay, so we have to describe the importance of meiosis. Remember, we said, I've mentioned it in the video also before, it helps maintain a constant total number of chromosomes from generation to generation. And this is an extra question for you guys. We've said it before. In meiosis, does the number of chromosomes stays the same or is it halved, or is it doubled? What happens? It is halved. Perfect. Okay, so the number of it becomes half. Yes. Okay, so you have to describe how the genetic code for making proteins gets from the DNA in the nucleus to the ribosomes in the cytoplasm. So which of the following accurately describes how the genetic code for making a protein gets from the DNA in the nucleus to the ribosomes in the cytoplasm? Okay, so the first one, a molecule called DNA is made by copying the mRNA code. That is false. It should be flipped. A molecule, not even by copying, but from DNA we get RNA, not from RNA we get DNA. mRNA is used to carry the information from the DNA in the nucleus to the mitochondria and the cytoplasm. Where did we say protein synthesis occurs? In the ribosomes. So if it was to the ribosomes in the cytoplasm, it would be correct. So the third one, the mRNA passes through the ribosomes which assemble the amino acids into protein molecules. That is correct. And remember we said the function of the tRNA is what? Function of tRNA is to bring these amino acids. Instead of copying, we say translating. Here, for it to be translating, we would say a molecule called the mRNA is made not by translating, by transcribing DNA. Translation is for the production of proteins. That's when we go from mRNA to proteins. Now the fourth option, the specific order of amino acids is determined by the sequence of bases in the mRNA. That's correct. Remember we said it's determined by the nucleotides or by the codons and not by the monosaccharides. What are stem cells? We already said undifferentiated cells that can produce specialized cells, and all the other options are wrong. What is gene expression? It's the use of genetic information to produce proteins. Okay? And remember we said all our cells contain the same set of genes. However, the way they are expressed is going to be different depending on their function, their location, okay? But they all contain the same set of genes. Okay, meiosis preserves the number of chromosomes in the zygote. True, when the male gamete and female gamete fuse, they form a haploid cell. That is false. They produce a diploid cell. Okay, remember, because haploid form a diploid. Each gamete must have a full set of chromosomes so that the zygote will have one complete set of chromosomes. That is false. We said the gametes have half the number of chromosomes. The halving of the number of chromosomes that occurs in meiosis is essential. Of course, it is essential. That's one of the main functions of meiosis, is to maintain a constant total number of chromosomes from generation to generation. Meiosis also helps increase the genetic variation. Yes, we said that before, by crossing over and by independent assortment, and that's the last option. The independent assortment of chromosomes and crossing over create genetic variation during meiosis, and that is true. Okay, we already spoke about stem cells. What are they? Unspecialized. They divide. What process? We said mitosis, to produce daughter cells that can become specialized for specific functions. We said there are two sets, embryonic and adult. The early stage, of course, is going to be the embryonic, and they have a huge potential, so they have a wider range, while the adult stem cells, the range is more limited, and the way they differentiate depends on the nature of the tissues these cells are found in. And we gave the example of the cells in the bone marrow. They can only differentiate into blood cells and white blood cells. Okay? Okay, I think that was your question, Tala. A molecule called mRNA is made by copying the DNA code. So here, by copying, they mean transcribing, okay? Not translating, watch out for it. Okay? It is used to carry the information from the DNA in the nucleus to what? To the ribosomes, because that's where protein synthesis occurs. The mRNA passes through the ribosomes, which assemble the amino acids into protein molecules. Okay, so we translate what the mRNA, so the ribosome binds to what? To the mRNA, and we said the specific order of amino acid is determined by the sequence of bases, okay? By the sequence of the nucleotides. Okay, during fertilization, what did we say? We said we have a haploid male gamete and a haploid female gamete, and they fuse and they give rise to a diploid zygote, okay? We said a segment of a DNA directly, whenever you see a portion, fragment of DNA, it's directly a gene, okay? Okay, crossing over. Whenever you see exchange of genetic material, it's going to be crossing over, and they're telling you it occurs in meiosis 1, specifically it occurs in prophase 1. And always remember, crossing over and independent assortment cause genetic variation. Here, independent assortment, when you see the word independent, it's random. They're not depending on each other. It's not because of this happens. It's completely random. When something is completely random, it's independent. So this is a way for you guys to memorize them. Okay, we said diploid cells contain 2 sets of chromosomes. Haploid cells contain 1. The diploid body cells contain 46 paired chromosomes. Okay, here they're talking about chromosomes, okay, not pairs. If they're talking about pairs, we say 23 pairs, not 23 chromosomes. Watch out for that. They might play on it. And if you see pairs of chromosomes, it's 23 pairs. However, if it's just chromosomes, it's going to be 46 chromosomes. Okay, so in mitosis, remember, if you haven't watched my video about mitosis and meiosis, make sure to watch them. Great explanation, and it's very short. So here they're telling us represent the structure of a chromosome during cell division. So remember I said in the video that before the cell is able to divide, it must duplicate or replicate its DNA. Okay, so once it replicates the DNA, instead of 1, there is 2. So that's why we chose the second image, and the process doubles its genetic material. We call it replication. And what is true about sister chromatids, they carry the same allele for each gene, and they are held together by a centromere. You see, this middle part is what we call a centromere, and they separate during mitosis. So remember, in anaphase, we said we have the separation of sister chromatids. Okay? Let's go next. Okay, here we have a nucleus of a body cell. How many chromosomes? It's straightforward. Which type does this cell most probably undergo to give sperm cells? So because they said sperm cells, we're talking about gametes, so of course it's going to be meiosis. And since we're getting meiosis, what's going to happen to the number of chromosomes we mentioned before? It's going to be halved. So since we have 4, we're going to select the images that have 2. Okay? So that's why we choose 1, 2, and the last one. Okay? If there's anything you don't understand, just stop me and I'll explain. Here they're just telling us to explain the process. So in the first one, you see different chromosomes and there's a different separation. It's random, that's why it's independent assortment. Here you can see there's an exchange of information, so we call it crossing over. And what is the function of them? It increases genetic variation and they create new combinations of alleles in gametes. Okay? Okay, this was the genetic code table I was talking about. That's just one you don't have to memorize. Okay? So here, let us see what they're telling us. Drag and drop the correct triplets to obtain the mRNA sequence based on this fragment of DNA. Okay, so since we have this shorts, the sequence below shows a part of a human gene. Okay, so they gave us the non-coding strand. Okay, so that's why from this we got Instead, what pairs with T, A? What pairs with A, U? Why U? Because we're looking at mRNA. So whenever we're talking about DNA, then four nucleotides are what? A, T, C, G. But when we're talking about RNA molecules, the nucleotides are going to be AU and CG. Okay? That's why instead of AT, I put U. And what pairs with CG? And so on. That's how we do all the rest. Okay? And here they're telling you to, from the genetic code, remember we said, what determines the sequence of amino acids? It is the sequence of bases. So as we saw, these are the sequence of bases AUG, UCC, ACU. These determine the amino acids. Okay? And we just look at the table. So AUG, we look at the table. Where is AUG? It's going to be here. So that's why it's met. And then UCC, you have to look in the table. Serine, SER. And then again, ACU, and so on. Okay? It's straightforward. Undifferentiated, unspecialized, directly. Don't even need to continue. They are stem cells. Okay, what is involved in sexual reproduction? Meiosis produces genetically identical. It's going to be mitosis, results in haploid cells. Of course, meiosis needed to replace worn out cells, mitosis, and it involves body cells, mitosis. So this is to go back to the question, I think it was number 2. That's why we said number 5 was false, because meiosis does not involve body cells, it involves haploid cells or sex cells. Okay? Okay, because they're telling us to synthesize proteins from the nucleus to the cytoplasm, it's going to be mRNA. Whenever you see in attaching the correct amino acids, it's directly going to be tRNA. Okay, these are the steps for protein synthesis. First, we said, as you saw in the image, the DNA split into 2 parts, okay? And then a copy of a gene is produced in the form of the mRNA. After the mRNA is produced, it leaves the nucleus, goes to the ribosome in the cytoplasm, and then we have amino acids from the cytoplasm. Who brings those amino acids? We said it was the tRNA, and it brings them based on the code carried by the mRNA with the help of the tRNA. Okay? Okay, so since they undergo meiosis, what's going to happen to the number of chromosomes? It's going to be from 46, we're going to half it. Half of 46 is 23. Same thing for the females, and then they fuse. Once they undergo fertilization, fertilization is basically the fusion of the male gamete with the female gamete. That's why we get back the 46 chromosomes. Okay, if an elephant cell has 56 chromosomes, how many chromosomes would the parent cell have at the beginning of the first mitotic division? It's going to have 56, however, what? Each with 2 chromatids. Okay, this is important to note. At the end of meiosis 1, they're going to have we're going to halve the number of chromosomes, because remember, meiosis 1 is the separation of homologous chromosomes. That's why we have 28 chromosomes, each with 2 chromatids, while meiosis 2 is the separation of sister chromatids. So we're going to have 28 chromosomes, each with 1 chromatid. Okay, during DNA duplication, or we also call it replication, each chromosome becomes a pair of 2 identical strands, now each called a chromatid, joined together near its centers by a centromere, as we mentioned before. Again, what determines the sequence of amino acids? It is the sequence of nucleotides. If a body cell contains 46 chromosomes, female gametes, of course, are going to have half the number, so 23, and a human zygote will have 46. Okay, this is just to show the stages. It's straightforward. You have prophase, metaphase, and they directly skip to the 2 identical daughter cells. While here you can easily tell, since there's 2 divisions, of course it's going to be meiosis, and since there's 1 division only, it's going to be mitosis. And in meiosis, remember, we have 4 cells, half the number of chromosomes, while in mitosis they are identical to the parent cell. Okay, number 30 now. This is just labeling. Okay, so we said when we have two sister chromatids joined by centromere, DNA duplication has occurred, and then this is the process of mitosis. And we already said the three main functions of mitosis. This is important. It allows an organism to reproduce through vegetative reproduction. It's usually for plants. Okay. This is also straightforward. Maintains original chromosome number, mitosis. Halves the number directly, meiosis. Diploid cells, mitosis. Gametes, meiosis. Takes place in most body tissues. Think about it logically. Body cells perform what? Mitosis. Our body cells are spread everywhere in our body. Okay? Takes place only in the ovaries and testes in humans. It's going to be meiosis. Occurs in one division, mitosis. Two divisions, meiosis. Again, for mitosis and meiosis, please watch the video. It's going to really help you, and it's very short. Okay, so how did I know this is protein synthesis? You can see this G is the tRNA. It's going to bring amino acids, and the E is the ribosome, F is the mRNA, so I directly know this is protein synthesis. So G is the amino acid. Okay, the tRNA is C. Yeah, because here they're showing us this is the one labeled. Here it's just the green part which is labeled. A is going to be the DNA. Okay, B is going to be the mRNA. C, we already said, is the tRNA. D is going to be what? The protein that's being produced. And E is going to be the ribosome. Okay, and F is also going to be the mRNA, while G, we said already, is the amino acid. So it's just a straightforward labeling. Okay. Again, this is the same thing. You have the ribosome, you have the mRNA, you have the tRNA, and this is the protein being formed. Again, what carries the amino acids? It's going to be the tRNA. Okay. How many pairs of chromosomes does this organism have in his cells? So pairs, every pair is 2, so 1, 2, 3, 4 pairs. Okay? Used to describe the chromosomes of the same pair. Remember we said homologous chromosomes. And how many chromosomes with the X cell? X cell. So what are we going to perform? Meiosis and not mitosis. And through meiosis, what happens to the number of chromosomes? It halves. So here we have 8, so we're going to have only 4 chromosomes. If they asked how many pairs, it's going to be 2 pairs, because we had 4 pairs, half of 4 is going to be 2. Okay, this is also straightforward. 2 sets, diploid. Skin cells, so our body cells, of course, diploid, includes gamete, it's going to be haploid. Once chromosomes are paired, diploid, 2 sets, of course, diploid, formed by mitosis, of course, diploid, and also diploid is always 2n equals to 46, so diploid cell. Okay, so we said when we're born, or when we're still an embryo, we have embryonic stem cells. As we become older, these disappear, and we have some adult stem cells, as they're telling us here, and we said again they divide by mitosis, but the range they have to differentiate and specialize is less than that of embryonic stem cells. Okay, I've also mentioned this, pay attention to it. Every 3 bases code for a particular amino acid. 3 bases, okay? So 3 bases make a codon, and we saw the genetic table, it was all 3, 3, 3, 3. So 3 codons determine the amino acid sequence. Okay, we said DNA is what? Genetic material that stores what? source information to make proteins. The sequence in which the nitrogen bases are arranged is very important because it forms the genetic code that is used to determine the sequence of amino acids in the protein that make up our cells. Okay, this is also straightforward. Again, because they told us in DNA, adenine makes the base pair with thymine while guanine makes cytosine. Let's say the question was how do the bases pair up in RNA? Adenine would pair up with what? Who's gonna tell me in the chat? Anyone? Okay, so if it was an RNA, adenine would make a base pair with uracil, which is U, while guanine will still make a pair with cytosine. The question was just, if the question was reversed to how do the bases pair up in DNA, I said adenine would make a base pair with uracil, which is U, while guanine would make a base pair with cytosine, which is C. Okay, so meiosis is what type of division? It's a reduction division because the chromosome number is halved from diploid to haploid. And that's it. I repeated the question. The question was just over here. If we were asked how do the bases pair up in DNA, in RNA instead of in DNA, the answer would just be, for the first one, adenine would make a base pair with uracil, while guanine would make a base pair with cytosine. And that's it for the quiz. It's pretty straightforward. If you have any questions or if I went over something quickly, let me know so I can explain it better for you. However, if it's all clear, just let me know in the chat that you understood everything and all is good. You're most welcome. Thank you all for joining. I hope it was beneficial and you're ready for your exam tomorrow. So, best of luck on your exam tomorrow. I hope you're gonna, all of you, ace your exam tomorrow, get 100. If you have any questions, let me know. And thank you again for joining. Best of luck and see you guys.